Hello, we are going to continue our unit on genetics. If you remember that genetics is the branch of biology that seeks to explain biological diversity. Here's a little bit of history and how it all came about, just so you know that things have really changed and a lot of the ideas that we've had have been rejected as more technology has come about. Okay? So we first started out with an attempt to explain the concept of inheritance, and they called that panagenesis. All right, well, Aristotle rejected it, okay, and that's not what happened. So Mendel was the guy who kind of came about and did all this and showed us the inheritance probabilities and possibilities when he did his research on pea plants. And again, remember, pea plants are very easy to grow. Very large numbers of offspring are produced. And these are the 7 different characteristics that he did as far as his research on. And we're going to do a lot of different examples with Punnett squares when we talk about this. When we talk about gene and allele, okay, the gene is the actual like type, like seed color, and the allele is the alternate form of the gene. So for seed color, it would be green or yellow. So the gene is the actual location and what it would code for, and the allele are the different types. Again, just showing you that gene location and then the different alleles. Genotypes are the genetic makeup or the letters that go with it, where the phenotype is the physical makeup of what it codes for. Okay, we'll show you examples of what a monohybrid cross is with doing Punnett squares, but it's a cross involving one trait. A test cross is crossing an unknown genotype with the known genotype. So the unknown genotype is always going to be the dominant. So is it going to be capital capital or capital lowercase? And we're always going to cross it with the unknown. And you get 2 different options. If you cross it with the total homozygous, then they're all purple. And if you cross it with the heterozygous, you get half purple, half white. So a test cross is the mating between unknown genotype with the homozygous recessive or the known genotype. Okay, the next type of Punnett square is called a dihybrid cross. So we're crossing 2 different traits and we get a bigger Punnett square. We get 16 squares. Okay. We also have a trihybrid cross when you have 3 different characteristics. And you get a 64 square Punnett square, which is insane. But you can figure out the probability by just multiplying the probability of taking each trait. And then you would be able to figure that out. All right. Pleiotropy is the idea that most genes influence multiple characteristics. So pretty multiple effects on a single gene. And the example we have is sickle cell anemia. The same allele causes both the abnormal pigmentation and a cross-eyed condition in tigers. So there are 2 different examples. We have sickle cell anemia and we have something in tigers. This is just showing you the sickle cell anemia. There's a lot that occurs because of the sickle cell gene. And again, the bottom here is the sickle celled. Epistasis is a gene at one location is going to alter the phenotypic expression of a second gene at another location. And what we see is coat color in mice. Pretty much black is dominant to brown, but there's another gene that is going to say whether the pigment will appear or not appear. And that's how you can get albinism to occur. So again, there's 2 different genes and one is going to affect the phenotype of the other gene. Again, just showing you with horses. Polygenic inheritance is an additive effect that occurs with 2 or more genes on a single phenotypic characteristic. And what we get is human skin color. There's about 3 genes that will add in for human skin color. And what you get is almost like a bell curve. And again, this would be that trihybrid problem that I had told you with 64 squares. Linked genes are genes that are located close together on the same chromosome. If they're linked close together, then they are tending to be inherited together. They don't typically follow that law of independent assortment. We do have some recombination or crossing over that occurs. And pretty much the further apart two genes are, the higher the probability that a crossing over will occur. So all phenotypes are subject to modification by the environment. So if you're supposed to be tall but you don't get enough nutrition, you're not necessarily going to be tall. And the same thing that happens with Siamese cats. Okay? All right. Multifactorial expressions are controlled by at least several genes and environmental factors. So eye color, hair color, height, weight, intelligence, skin color, heart disease, diabetes, cancer, alcoholism, spina bifida, cleft palate are all things that are multifactorial. So if you have any questions, please let me know. We're going to be practicing tons of Punnett squares, and we're going to be doing lots of problems to understand. Thanks.